Hello, so we're ready for the next part of our notes on cells. So last time we talked about the structures that all cells have in common and then prokaryotic cells. Now we're going to talk about eukaryotic cells and all of their associated organelles. So the hallmark of eukaryotic cells is that they have compartmentalization. They have separation inside their cytoplasm. One of those pieces is a nucleus, and the nucleus has a double membrane around it, and it's going to contain the DNA. Another thing that eukaryotic cells have is a cytoskeleton that provides support. So let's talk about those organelles. So first we have the nucleus, which again holds the genetic information. Now in earlier grades, you might have been told like the nucleus is the control center of the cell. I want you to stop thinking about it like that because it doesn't control anything. It's just a repository, a place for the genetic information to be held. The nuclear membrane, that double membrane that has pores in it, allows certain things to go into and out of the nucleus to have access to the DNA, but the nucleus itself does not control anything. Okay, the DNA directs the making of proteins, but that's still not controlling anything. Most cells have a single nucleus. Some cells will have multiple nuclei, because if the cell is a little bit bigger, then it might need more nuclei to have more DNA to be able to actually help it make more proteins and things like that. Inside of the nucleus is the nucleolus, and this is a little structure that is going to synthesize ribosomal RNA. So one of the types of RNA that makes up the ribosomes. Then we have the nuclear envelope, or like the nuclear membrane, like I said, which has 2 phospholipid bilayers. Okay, those ribosomes, that ribosomal RNA, which is where the protein synthesis is going to take place. Okay, so all cell types, even the prokaryotes, have these ribosomes. A ribosome is a complex of some RNA, we call it rRNA, and some proteins. So there's different subunits to that. There are basically 2 places that you're going to find ribosomes, and that's free floating in the cytoplasm, or they're going to be associated with other membranes like the endoplasmic reticulum that are in the cell. And depending on where they're located, usually goes to what function their proteins that they're assembling will do, or where those proteins will stay. Okay, then we have the endomembrane system, which is a large series of membranes that runs throughout the cytoplasm, and it separates the cell into different areas. Each of those membranes has different cellular functions where different things will occur. And this is that huge distinction between our cells, animal and plant and fungus cells, and the prokaryotes, or the bacteria cells. They don't have any of this internal compartmentalization or membranes. One of those pieces is something called the endoplasmic reticulum. There's 2 kinds of endoplasmic reticulum, the rough and the smooth. The rough endoplasmic reticulum, or RER, has ribosomes covering its surface, and that's why it has that rough appearance to it. This is where proteins will be made that are secreted or sent out. So those excuse me. Sorry about my dog. Those are the proteins that are going to be sent to the lysosomes or to the plasma membrane. So the rough ER, those are the ones that are going to leave. They're going to go. They got to get out. Okay. Then we have the smooth ER, smooth endoplasmic reticulum, or SER. And that doesn't really have very many ribosomes on it at all. And it does have a lot of different functions. So the smooth ER does things like put together or synthesizes lipids, will store calcium. It's one of the biggest things it does is detoxification. So you're going to have a lot of smooth ER in organs like your liver. Okay. And the ratio, so how much of the rough ER to the smooth ER you have in each cell depends on what that cell needs to do. Then we have another membrane type organelle, and that's the Golgi Apparatus. It's also sometimes called Golgi complex or Golgi bodies. As long as you see Golgi, they're all talking about the same thing then. Okay, so the Golgi is flattened stacks of connected membranes. It kind of almost looks like a stack of pancakes that you've dropped some syrup on or something to that effect. There's lots of different things that people see when they look at that, but it's like a stack of membranes and it's like flat. Okay. The main purpose of the Golgi is to package and distribute things that were made at other parts of the cell. So if it's going to be made in one part and used in another, the Golgi is going to have a role in it because it's going to package it up and get it to where it needs to go. So the Golgi has two faces or two parts to it, the cis and the trans face. And so what's going to happen is that on the cis face is where things are going to enter. So here, whatever product needs to be packaged up and distributed is going to enter, and then it's going to leave the trans face. So I kind of like try to remember it like trans face is where it's going to finally get transported. So it comes in the cis and it leaves the trans. Another way you can remember that is because cis just starts with a C and it comes first. It's where it goes in, and trans starts with a T and it is after. Okay. So here's just another diagram of the cis or receiving side. And then as the vesicle, whatever is going to happen, it gets processed. Excuse me, I'm having trouble today. And will form these little vesicles. So when you see these little bubbles that are like off of the Golgi, that's what's happening there. There's little vesicles that will leave to go to other areas of a cell or even to different cells. Okay. Then we have the lysosomes. The lysosomes are the digestive parts of the cell. They are little vesicles or sacs that come from the Golgi bodies or Golgi complex. They have enzymes in them, and those enzymes are going to help break down macromolecules that we eat. They're also going to help destroy cells that are no longer useful or foreign cells, foreign matter. Anything that the cell has taken in through that process of phagocytosis or cell eating, where it's wrapping around something with a membrane, wrapping it in a membrane, and then the lysosomes will help digest it. Okay, the next one we have are microbodies. Microbodies are enzyme-bearing and membrane-enclosed vesicles. So here's where we actually have a full lipid bilayer plasma membrane vesicle, unlike some of the other ones that we'll see. And inside of that vesicle are certain enzymes. So peroxisomes are one of the microbodies that we have to learn about. And they're good for the oxidation of fatty acids. That's their function. That's what they do. When they oxidize, oxidation is loss of electrons. They're pulling off electrons. Hydrogen peroxide is produced. And then that hydrogen peroxide, that's not good for our bodies. So we have to find a way to get rid of that. So that is made harmless to us by a catalase, an enzyme. So that's kind of what microbodies do. Then we have some other structures called vacuoles, which are bigger than vesicles. And vacuoles, you're going to find those in plants, and they're going to be storage structures for the most part. They have a lot of different jobs as like storing different materials. The central vacuole is going to be a warehouse for storing water. It's what helps the plant maintain its shape. And then we have the contractile vacuole in some fungi and protists. It's right there. When we do our lab on the paramecium and gizmos, you'll get to see this in operation and see what's happening and how the paramecium maintains homeostasis using its vacuole that actually like pumps. Okay. And then there's other storage vacuoles. Okay, moving on. We have the mitochondria, the one that everybody remembers as the mighty mitochondria. One thing that's really cool about mitochondria and chloroplasts is that they have their own DNA. It's completely separated, and it's inherited in us and other animals from the maternal line, because the egg is the bigger of the two. So eggs are bigger than sperm. They have all of the necessary cell organelles in them. So when the sperm fertilizes the egg, that's your mom's mitochondria that's in there. So those are going to be self-replicating, and that's what's going to continue. So you have your mom's mitochondria. So you have mitochondrial DNA. So mitochondria, that's where metabolism is carried out. So that's where we're going to turn our food, our glucose, into ATP energy. It has membranes. It has an outer membrane, and it has an inner membrane. And then the inner membrane has folds or creases in it, which we call cristae. Inside of that inner membrane is a matrix, and that's where a lot of the processes of metabolism are going to happen, which we'll talk about in our next unit. Then we have chloroplast, which is a type of plastid that contains green pigment or chlorophyll. Some other organisms have this besides plants, but it's mainly found in plants. So like a type of protist called a euglena will have some chloroplast in it. Okay. They contain chlorophyll, and again, we've got that double membrane system happening. Inside of the chloroplast are stacks of thylakoids, and that stack of thylakoids is called a grana. And this is where photosynthesis is going to happen. So this is how the sugars are produced in the plant. And again, we'll talk about that in another unit. Chloroplasts also have their DNA. So many scientists believe that chloroplasts and mitochondria were separate cells at one point in time and then got engulfed by another cell. Okay, and that's what this is. It's called the endosymbiosis theory. And so what scientists believe happened was that there were these free living cells, and you had a cell that was a cyanobacterium that had was photosynthetic, was able to produce its own food, and it kind of got inside of another cell, okay, another prokaryote, and it started to live in there and live in a symbiotic relationship where it wasn't being digested or eaten. The cyanobacteria got a nice place to live. The other prokaryote that engulfed it was able to get some food from the cyanobacteria that it was producing. And eventually, through time, as these aerobic bacteria and the cyanobacteria were inside the cell, and they started to replicate and make more mitochondria, more chloroplasts, and it became the first eukaryotic cell, okay, with a nucleus forming from folds that were created in the cell as these other organisms were moving in. So it's kind of somebody's moving into somebody else's house and then just decides to become part of them, part of their family. Okay. So here's just a diagram showing where they would have come from. So protobacterium became the mitochondria. Something else probably became the nucleus. Cyanobacteria became the chloroplast. Okay. So are we completely sure? No, because we weren't there. But there are evidences for it, as in the mitochondria and chloroplast both have their own DNA. So that's one of them. We'll talk about more endosymbiosis in a later chapter. Okay. We're almost done here. So the cytoskeleton is our next structure, which is a network of protein fibers that's going to help support and shape the cell. And it's going to help keep those organelles in fixed locations within the cell. It is a always changing system. It's constantly forming and pulling apart and reassembling in whatever the cell needs it to be. Okay.